Hello everyone, welcome to our presentation on coding the human genome, tools and technique in nutrigenomics. I am Leslie Ryan and today we will explore how cutting-edge genetic research is reshaping our understanding of nutrition, especially for conditions like type 2 diabetes mellitus. Let's start with the basis. Nutrigenomics is the science of how food communicate with our genes. It is two-way street. First, nutrients influence gene expression, turning genes on or off to affect metabolism, immunity, and more. Number two, your unique DNA determines how you respond to food. For example, FTO genes link to obesity. Certain variant like RS9939069 can increase appetite and belly fat. Meanwhile, the CETP gene affect HDL cholesterol level, which impact heart disease risk. This is not just a theory, it is the foundation of personalized nutrition. Imagine diet tailored to your DNA, helping prevent disease like type 2 diabetes mellitus. But why we focus on this disease? Let's look at the prevalence statistic. Type 2 diabetes mellitus is global crisis. 50% of adults are overweight or obese. A key risk factor, even worse, 60% of diabetes mellitus cases go undiagnosed, silently damaging organs. Between 2015 and 2019, diagnosed cases jumped from 13.4 to 18.3%. This is some of the factors that can driving this problem. First is lack of awareness. Second, limited screening. And three, poor lifestyle habit. But there is hope. Nutrigenomics tools like GWAS and metabolomics can spot risk early. For example, low magnesium levels are tied to insulin resistance, but genetic testing could flag this before the symptoms appear. Still, challenge remains. These tools are expensive and aren't standalone solution. Bridging this gap is critical to turning science into real-world health gain. And next, we will dive into the tool uncovering this gene-diet connection.
Next, we move on to why diabetes being held public concern. The first one because of the lack of awareness. Based on WHO, it states that in 2022, there are more than half people living with diabetes do not take medication for their diabetes. Second is lack of screening, which leads to increasement of diabetes cases. Based on WHO, the cases of diabetes has been increased from 200 million in 1990 to 839 million in 2022. Why most of people do not screen their health consistently? Maybe because them feeling health, and some people also could not afford the cost of medical checkup because nowadays it's a bit expensive for them. The last one, diabetes disease, is one of the highest that causes death in Malaysia. Based on WHO, it states that in 2021, diabetes and kidney disease due to diabetes causes over a million of deaths. There are four main objectives to be achieved in this topic. The first one is to introducing the genome-wide association studies, which is the GWAS. The second one to explaining the role of synapse. Third one is to exploring the application of omics-based tools. And the last one is to demonstrating personalized nutrition strategy for prevention and management of type 2 diabetes mellitus.
Hello everyone. Today I'll discuss the role of genome-wide association studies, GWAS, and omics in understanding genetics and health. My name is Michael O'Neill Glenn Amizai, and let's get started. GWAS identifies genetic variants linked to diseases by analyzing human genomes to find variants more frequent in affected individuals. Recent advancements in genotyping have improved our insights into conditions like obesity and diabetes. Microarrays detect gene expressions using a chip with nucleic acid fragments. RNA sequencing sequences all RNA in a sample for gene expression analysis. Mass spectrometry identifies and quantifies ions based on mass to charge ratios. NMR spectroscopy analyzes molecular structures in a magnetic field. GWAS reveals genetic variations related to dietary traits, identifying SNP linked to lactose intolerance and caffeine metabolism. Microarray show how dietary fat affect gene expression, while RNA sequencing analyzes responses to various diets. MS and MMR help understand how genetic impacts metabolism and lipid profiles. In summary, combining GWAS with omic technologies enhances our understanding of genetic and diet, paving the way for personalized health strategies. Thank you.
Hi everyone, now I will present about the genetic factor which have two, which is the FTO and CETP gene. So for the FTO, it is located at the 16th chromosome. So what is important SNP, which is RS9939609 and RS8050136. So what is the effect? It causes higher body weight and belly fat, and it also causes bigger appetite and early sign of madness in kids. Now I will tell about CETP gene. So the important SNP, which is RS708272. So what is the function? So it control good cholesterol, which is HDL. So with the version more HDL, then lower heart disease risk. So it linked to unhealthy blood fat, which is part of the madness.
Next, for the application, firstly, we have genome-wide association study GWAS that can identify diabetes-related gene that can detect genetic risk using single nucleotide polymerism SNPs, and also can support personalized diet planning. Secondly, we have metabolomics that can analyze blood or urine metabolites and can detect biomarker for insulin resistance, also can track diet response in real time. Thirdly, we have epigenomics that studies diet-induced DNA methylation and explain how early life nutrition affect type 2 diabetes risk later. And lastly, we have transcriptomic that measure gene expression changes through the diet and reveal how food affect insulin signaling and inflammation.
Hi, I will discuss about nutrient role, which is magnesium and disease mechanism. So basically, magnesium is a mineral that often get overlooked, but it's incredible importance, especially when it comes to blood sugar and insulin functions. So magnesium it help insulin do its job. So it allow insulin to move sugar or glucose from our body into the cell where it used for energy. So without the enough magnesium, this process doesn't work well. So insulin becomes less effective, and that can lead to insulin resistance, which are a key factor in the development of two types diabetes. So, low magnesium also means less sugar is able to enter the cell. So, as a result, sugar stays in the blood and the blood sugar level rise. Over time, this can put stress on the body and increase the risk of health complications. So, magnesium also protects the pancreas, especially the beta cell that produce insulin. So, when the magnesium level are low, this important cell can get damaged, reducing insulin production even further. And finally, magnesium helps fight inflammation and oxidative stress, where two things that are already increased in people with diabetes. Without enough magnesium, the body has harder time controlling this damage. In total, magnesium is involved in over 300 functions in body. Many of them relate to sugar metabolism and energy production. So if we want to support healthy blood sugar level and keep our insulin working properly, getting enough magnesium is essential. Thank you.
Some individuals who carry high-risk genetic variants may never develop the disease,
while others without unknown genetic variants may develop the disease due to the poor lifestyle habit. Genetic tools are complementary in helping refine risk prediction, but not serving as a standalone solution. Clinical assessment remains as the gold standard for predicting. Although health insurance coverage and subsidy paid by the government in some low-middle income countries, genetic services are high cost and remain a barrier, especially for the low-income and rural patients. In conclusion, nutrigenomics bridges the gap between genetics and nutrition by offering insights into how dietary components interact with genes to influence the risk and management of type 2 diabetes mellitus.